hi everyone, and welcome to Fueling Your Gut Reactions. My name is Samantha, and I'm a microbiology PhD student in Dr. Pamela Chang's lab at Cornell, where I study the gut microbiome, which is what we'll be learning a little bit about today. So we're going to kick things off by setting up our experiment. The reactions that we'll be observing do take some time to occur, so we'll set them up together now, and then we'll learn more about them and make some hypotheses. So just a quick note, I'm in my kitchen, if you can see, and even though we're setting up our reactions with ingredients that you might find in your kitchen, these are still science experiments. And when we're doing science in the lab or at home, we don't eat or drink anything that we're working with. So our reactions are going to be happening inside sandwich bags. So you'll need one for each reaction. We'll be looking at 7 different reactions. So there will be 7 bags in total. For each reaction, you're going to be adding the ingredients to the bag, pressing the air out of the bag carefully, sealing the bag, and mixing the ingredients in the sealed bag by pushing on the bag with your fingers. I'll demonstrate as we go along. You might want to get a Sharpie or another pen and label your bags now. You can just label them 1, 2, 3, 4, 5, 6, 7. So 1 through 7 to help you keep track of your experiments as you go. First, we need to start with some controls. And if you can measure out the ingredients that we're using exactly, that's great. If not, that works too. In that case, you might be using a plastic spoon or a small plastic cup that was provided to you. Starting with our positive control, we're going to add 1 teaspoon of yeast. And if you're using a plastic spoon, just use 1 spoonful. 1 spoon of table sugar. Again, just using a spoonful with your plastic spoon. And then 50 milliliters of warm water. If you're using a little plastic cup, that cup's going to be about 2/3 full. I'm using a measuring cup here, and I'm going to go ahead and pour that into my bag. So now I'm going to do my best to press the air out of this bag and then seal it up carefully. We don't want these to leak. Okay, and this is in bag number 1. This is a positive control because these are conditions under which we do expect our reaction to work. And we'll talk a little bit more about that in some upcoming slides. So bag number 2. For our negative control, again, we're going to be adding 1 teaspoon of our yeast. Again, that's just a spoonful. And again, 50 milliliters of warm water. No table sugar this time. Okay, I'm going to go ahead and add my water, try and press the air out, do the best you can there, and then smush that up with your fingers. Okay. And this is the negative control because these are conditions under which we do not expect our reaction to work. The positive and negative controls are going to give us a sort of baseline that we can compare the rest of our reactions to. For the rest of our reactions, we'll be changing up some of the ingredients a bit, and those changed ingredients will be our variables. The first variable that we're interested in seeing the effects of is nutrient availability. Here, this is reaction number 3. We're going to be adding, again, 1 teaspoon of our yeast. Again, that's a full spoonful. And again, our 50 milliliters of warm water. But this time, we're only going to be adding a pinch of table sugar, just a pinch, instead of a whole teaspoon. So this is going to mimic an environment where nutrients are limited. So we're going to set aside bag number 3. Our next variable is pH. Can anyone tell us what pH means? pH is a measure of how acidic or basic something is. So in bag number 4, we're going to set up an acidic environment for our yeast. We have bag number 4. We have our yeast. Take 1 teaspoon of our yeast. Take 1 teaspoon of our table sugar. But this time, instead of water, we're going to use 50 milliliters of vinegar. I'm going to go ahead and measure that out. If you're using a small cup, that's 2/3 of the way full. Okay, so let's get our vinegar added to bag number 4. Press the air out and seal that up tightly. Now, to make a basic or high pH environment for our yeast. In bag number 5, again, we're going to start with our 1 teaspoon of yeast, our 1 teaspoon of table sugar, our 50 milliliters of warm water again, but this time we're going to be adding an additional 1 teaspoon of baking soda. So let's get this bag sealed up, bag number 5, mix that around. So the pH of vinegar up here is about 2.5, which is what makes it acidic, compared to water, which is around 7. That's neutral. So other examples of acids are lemon juice and orange juice. Now, on the other hand, baking soda has a pH closer to 9, which makes it basic. Some examples of stronger bases that we're not going to use today are laundry detergent and bleach. Our last variable will be the type of nutrient provided to our yeast. So in bag number 6, again, I bet you can guess what comes first, 1 teaspoon of yeast. And this time, instead of table sugar and water, we're going to be adding 50 milliliters of milk. So I'm using just regular old milk here. You might have powdered milk that you're going to need to mix with water according to directions. Then you'll be going ahead and adding those 50 milliliters of that milk drink mix. So I'm adding my 50 milliliters of milk to my yeast, and that's bag number 6. Okay. Now in our last bag, bag number 7, once again, we're going to start with 1 teaspoon of our yeast, our 50 milliliters of milk once again, but this time we're also going to be adding a crushed up lactate tablet. So if you have a spoon, you can crush this with the back of your spoon and then open it up carefully. I already have mine crushed. And make sure you dump all that powder from that caplet into your bag the best you can. So now we're going to seal this last bag up, and we're going to set our reactions to the side. We'll check back in on them later. Great. So now we've got all our reactions set up. We do need to wait for a few minutes for them to occur. In the meantime, let's learn about what's happening in these reactions. And if you were wondering what our yeast looks like, this is a picture taken with a microscope of yeast, the microbe that is carrying out the reactions that we just set up. I'm a microbiologist, and today we'll be talking about microbiology. But what does that mean? Does anybody have a definition for microbiology that they'd like to share? Microbiology is the branch of biology dealing with the structure, function, uses, and modes of existence of microscopic organisms, also known as microbes. So what are microbes? The word microbe comes from the Greek language. Micro meaning small and bios meaning life. This term refers to microscopic organisms, often too small to be seen with the naked eye, and includes bacteria, fungi like our yeast, as well as protozoa and viruses. During our workshop today, we'll mostly be talking about bacteria and of course fungi, those yeast that we just used to set up all our reactions. So what are some of the reactions these microbes can carry out? We can first learn about the reaction that's happening in our bags right now, and that's called alcoholic fermentation. Our yeast can break down sucrose, which is just the scientific name for table sugar. And when they break it down into glucose and fructose. So glucose is a very important simple sugar that is used for energy by many living organisms. We use it, and so do our yeast. So when we pressed the air out of our bag, we were trying to get all the oxygen out of the bag and create what's known as an anaerobic environment, an environment without oxygen. In this anaerobic environment without oxygen, the yeast can break that glucose down to make ATP, a small molecule that's also very important for energy, during a process called glycolysis. That's depicted in this yellow box. But today, what we're interested in is what happens to the leftovers from this reaction. The 2-pyruvate leftover can be further broken down during that alcoholic fermentation to produce a little more energy, and the gas carbon dioxide, so known as CO2, and alcohol, specifically ethanol. With this background knowledge, we can start to make some hypotheses about the experiments that we just set up. A hypothesis is an educated guess about what will happen in your experiment. It's not necessarily about being right or wrong, but making a guess based on what you know, testing it with an experiment like we're doing today, and then learning from the results. So you can test your hypothesis by observing what happens in your experiment. So with our sense of sight, we can tell if carbon dioxide, that's CO2, is being produced because it's a gas. We might be able to see it as it forms bubbles. Think about the bubbles in your soda. Those are also CO2. Or as it causes the bag to expand. And with our sense of smell, we can tell if ethanol is being produced. Ethanol is a type of alcohol, and it might smell a little bit sweet to you. It might also remind you of something else that we're using a lot of lately. Does anyone know the products that I'm thinking of? You can just shout it out. The CDC recommends that it contains at least 60% ethanol. So ethanol is also found in hand sanitizer, like you can see on this label here. So another name for ethanol is ethyl alcohol. You might see that name on the label like I'm showing you here. Let's think first about our controls. These are the experimental setups that should produce an expected result. They tell us that our experiment is working correctly. So for our positive control, we set up our experiment so that it should work. So in the positive control bag, we have yeast, sugar, and water. What do you think will happen? What do you think you're going to observe in the bag? You can start to write these things down on your worksheet under hypothesis. So even though we know what should happen, we can record this as a hypothesis on our worksheet. My hypothesis is that the yeast are going to break down the sugar to make CO2 and ethanol. I'm going to predict the results for now, but we're going to wait to record those observations until we can actually see them. So I'm going to predict that bubbles will be produced, the bag might expand, and we might be able to smell ethanol. For our negative control, we set up our experiment so that it should not work. In the negative control bag, with only yeast and water, no sugar, what do you think will happen? What do you expect to observe in the bag? Again, though we know what should happen, we can record this as a hypothesis on our worksheet. My hypothesis, that yeast has nothing to break down, I think we'll observe no bubbles, the bag won't expand, and we won't smell anything new. So again, only fill in your hypothesis for now, and we'll have to wait a little bit longer to observe our bag, and then we can see if those hypotheses are correct. Moving on to our first variable of nutrient availability. If we only gave the yeast a pinch of sugar instead of a full spoonful, what do you think will happen? What do you expect to observe in the bag? Does anybody have any guesses that they want to share? And when we think about our variable of pH, if we put the yeast in an acidic environment instead of a neutral environment, what do you think will happen? Do you think the yeast can still work and break down sugar if they're in an acidic environment? Think about what you expect to observe in the bag and record your hypothesis. And if we put the yeast in a basic environment, again, instead of a neutral environment, what do you think will happen? Do you think the yeast can still work and break down sugar now in a basic environment? And what do you expect to observe in this bag? Write your hypotheses down now, and we can come back to them later. You'll notice that I haven't mentioned the bags with the milk yet, and that's because those reactions are actually a little bit different. The type of sugar found in milk is called lactose, and it's different from table sugar, that was sucrose, but it can also be broken down into, you guessed it, glucose. However, it requires an enzyme, and an enzyme is just a protein that helps make a chemical reaction happen, called lactase. Our bodies can make lactase and break down lactose, but sometimes our bodies aren't very good at this. And if we don't make enough lactase to break down the lactose we consume, this can result in someone having a certain medical condition. Does anybody know what that's called? That's someone being lactose intolerant. So maybe you know someone who's lactose intolerant, or maybe even you're lactose intolerant. People that are lactose intolerant can take medicines like Lactaid that contain this lactase enzyme to help them break down the lactose they might consume when they drink milk or eat things like cheese. Some bacteria can also make lactase and break down lactose. Today, we'll find out if yeast can do this or not. So if lactase is present, whatever we're looking at, bacteria, maybe yeast, can break down lactose into glucose. That glucose goes through glycolysis again, and then alcoholic fermentation can occur, ultimately producing CO2 and ethanol. So when our variable is the type of nutrient we give our yeast, this time lactose, milk instead of table sugar, what do you think will happen? What do you expect to observe in the bag? Do you think yeast can use lactose as a nutrient source? I'm asking you to take a bit more of a leap here and come up with a guess, and we'll all be excited to see what happens at the end of the experiment. And in our last bag, if we're giving our yeast both lactose and lactate, that lactase enzyme in the form of a lactate pill, what do you think will happen? What do you expect to observe in the bag? We're going to wait a little bit longer to check in on our reactions and learn a little more in the meantime. So these reactions might remind you of a popular quarantine cooking activity. Does anybody have any guesses what I'm thinking about? I'm thinking about making sourdough bread. To make sourdough bread at home, you get what is known as a starter. Starter might be a liquid batter or a stiff dough made from flour. Flour naturally contains a variety of yeast and bacteria. It's the yeast in the starter that can carry out alcoholic fermentation, and just like we will see in our bags, produce ethanol and CO2. Here, the CO2 is the really important product. Producing CO2 gas makes bubbles in the dough, like you can see here, and it allows it to rise, which is a process called leavening. This is why there are little holes or empty pockets in your sourdough bread, even if you don't make it yourself and you buy it at the store. Now, you might have noticed that sourdough bread tastes a little tangy or sour, like the name sourdough implies. But what puts the sour in sourdough? It's anaerobic fermentation again, but this time the pyruvate produced during glycolysis will get broken down by bacteria that naturally occur in the flour of the sourdough starter. Bacteria-like members of the Lactobacilli genus can take this pyruvate, make a little bit of energy, and now they're making lactic acid. So it's this lactic acid produced by bacteria during lactic acid fermentation that makes the dough acidic. So it would have a lower pH and contributes to the sour taste in sourdough bread. Lactic acid fermentation helps to produce some other foods. Can anyone think of any? One food that I'm thinking about is yogurt. So what's happening when we've got lactic acid fermentation carried out by bacteria to make yogurt? Bacteria have this lactase enzyme that can take the lactose in milk and break it down into glucose. Then they carry out that lactic acid fermentation to help produce yogurt. There's two really important types of bacteria that help to make yogurt. One is Streptococcus thermophilus, and the other is Lactobacillus bulgaricus. These two types of bacteria break down the sugar in milk, that lactose, and make lactic acid through lactic acid fermentation. This is what makes yogurt slightly acidic and why it has a tangy, kind of sour taste. If you happen to have a yogurt container with you, or maybe the next time you're at the store, you can look at the label. You see names of bacteria and the ingredients. I have a container here, and I can see S. thermophilus, L. bulgaricus, those bacteria that are on your screen right now, and L. acidophilus, L. lactis, L. plantarum. Those are all lactobacilli species. Sometimes people refer to these helpful bacteria as probiotics. And you might see this word on your yogurt container too. I know I see it. These types of bacteria are very helpful, not only in making yogurt, but also when they live and work in our intestines. So when we talk about the bacteria living in our intestines, we're talking about the gut microbiome. The term gut microbiome refers to the community of microbes that live in our gut, in our stomach, our small intestine, and in our large intestine. intestine. But how do these microbes make it into our body? We first come into contact with these microbes when we're born, and then our gut microbiome develops and changes as we start to eat solid food. And we know that some types of food also contain probiotics. We might also be exposed to different microbes in our environment, which changes over time. Microbes can also be found in drinking water, and we can be exposed to other people's microbes, some of which can make us sick, like if we get sneezed on. Your gut specifically is home to trillions of microbes right now, and they're influenced by some of the things that we've been talking about today. The microbes in our gut need nutrients to live. They get these nutrients from the food that we eat. If we're providing them with a healthy diet and the nutrients that they need, these microbes will grow happily in our gut. The gut microbiome is also influenced by pH. The stomach acid in your stomach has a low pH. So the parts of your digestive system, the parts of your small intestine, closer to the stomach are more acidic than the parts that are farther away. That's your large intestine or your colon. Not all microbes can live in this acidic environment, but bacteria like lactobacilli in yogurt can. In this picture, they're depicted as the green bacteria. If you have lactobacilli in your gut microbiome, they can help you digest lactose from milk, just like they do when they help to make yogurt. Scientists around the world, like me, study the effects of gut microbes and how they affect human and animal health. We know that helpful gut microbes like lactobacilli not only help us break down foods, they can also help us make vitamins that our body can't make on its own. They can help to protect us from damage to our guts during diseases like inflammatory bowel disease or IBD. And they can even protect us from bad bacteria called pathogens, like salmonella that can cause food poisoning. So now we get to be scientists together, even if we're in our kitchen, and look at the results from our experiments today. So it's time to observe our bags and see if our hypotheses are correct. We can record our observations on our worksheets. So we'll look at what do we see, what do we smell, and do our results support our hypotheses. I did have a table here, but it's gone. That's okay. So we're going to start with bag number 1. So this is your bag, your positive control with yeast and sugar and water. So hopefully you can see my bag expanded quite a lot, and it has a lot of bubbles. So bubbles, check. Bag expanded, check. Now, if you're careful, you can snip the edge of your bag or open it up and see if you can smell ethanol. I can smell ethanol. So check, check, check. We made CO2 and we made ethanol. So that's our positive control. That means our experiment is working the way we expected. Next, we'll move on to bag number 2. And that was our negative control. So yeast and water, no sugar. So sure enough, here's my bag, did not expand. And I don't really see any bubbles. I think the only bubbles in there are ones that I introduced by mushing the bag up. And if I smell it, I don't smell any ethanol. So no bubbles, the bag did not expand, and I don't smell ethanol. So again, the reaction didn't work, but that's what we expected to happen because it's our negative control. We can get into our first variability, our less sugar bag, and that's bag number 3. Now, we said that maybe this bag won't expand as much. You won't see as many bubbles. So I can look at this bag. I do see some bubbles. It expanded a little bit if we compared it to our negative control and to our positive control. So here's bag number 3 compared to bag number 2. It expanded a little bit, but not as much as bag number 1. This is bag number 1. Okay, so in a nutrient-limited environment, these yeast don't have as much of an energy source as enough food to work with, so they're not going to be able to carry out as much alcoholic fermentation. We can look at our pH variable now, starting with bag number 4. Now, maybe you predicted that yeast would not be able to function in this acidic environment with vinegar. If you said that, you were probably correct. So there's no bubbles at all in this bag. The bag did not expand, and I don't smell any ethanol. So sure enough, yeast are not super tolerant of a low pH. So they're not going to be happy here. They're not going to grow and carry out any of those reactions. So if these yeast were in your intestine, they would be farther down in your colon, in your large intestine. They would not be in your small intestine close to your acidic stomach acid. We can look at bag number 5. We're looking at our basic bag. I might have gotten these out of order, but we're looking at our basic bag now. And in this case, we do see some bubbles, but not as many as in our positive control. Didn't expand quite as much. So we learned here that at least in a basic environment, an environment about pH 9, the yeast can survive and carry out some of our reactions, some of our alcoholic fermentation. Now we're going to get into our last 2 bags, and these are 6 and 7. And these are the bags that I'm really excited about. So bag number 6 is our bag with yeast and milk. So we have lactose as our energy source here. So I don't see any bubbles. The bag did not expand, and I do not smell ethanol. So no CO2 was produced and no ethanol was produced. So yeast on their own cannot break down that lactose. So they must not have lactase. And sure enough, I know that they don't. They were not able to carry out alcoholic fermentation. But you can look at our last bag, bag number 7, and we do see some bubbles. Now, definitely not as many as in our positive control or some of our other bags, but they are there. The bag did expand a little bit. I can smell a little bit of that ethanol smell. So what's our conclusion here? Does anybody want to chime in? So when we get to this very last bag, where we give our yeast both lactose, which they can't digest on their own, we saw that with bag 6, we give them that lactate pill, that lactase. Now that lactase that we gave them is going to break the lactose down for them. Now that they have that glucose made available, now they can carry out alcoholic fermentation. And that's why we see bubbles, the bag expanding a little bit, and we can smell it a little bit as well. All right, awesome. So we can think about, did our hypotheses match what we observed? And it's okay if they didn't. Hopefully you learned something along the way. And that's what science is all about, making these hypotheses, carrying out experiments, and then learning from the results. And then sometimes you have to ask another question, go through the process again. So hopefully you learned a little bit about making hypotheses, a little bit about fermentation reactions, and what yeast and bacteria do when they carry out these reactions, both in foods that we consume and within our intestines. And Helena helped us with a really nice conclusion at the end there. So I would like to thank you for coming to my workshop today. And if you got to come to our workshop in person, you'd see our entire lab. We're the Chang lab up at the vet school at Cornell.